Bryce Maricco, Total Acre, manager of trial data and analytics. Sitting to my left is Chad Becker with Mosaic, and to my right is Brian Herbek with W&J Farms. We're in Nebraska today looking at residue trials. Liking what we're seeing so far, but really want to have a discussion today about how this product could fit into management plans here for growers in Nebraska, especially high yield growers with Total Acre like Brian Herbek. So Brian, just a quick question. What does residue management look like for you? Fall, spring, are you using any products? What gets you excited about an enzyme-based residue digester? What does that look like for you?
We're looking at something that we can put on the farm. The easeability of it, being able to mix it with an application that we're already doing. Right now, we know that dealing with the trash or the residue, it's a big thing with this wind that we've been having and dry weather. We can't get it to break down. It moves all over. So we really need to start getting it, getting it broke down and getting it to get stuck in the dirt. So.
Absolutely. Yeah, we were talking a little bit before. I know y'all been dry here for a few weeks now. We did bring the rain with us. We traveled here and brought the rain and got the car stuck quite a few times already. Could just cover it in mud nonstop. But, talking a little bit about the humectant in the product, trapping moisture, I, in my mind, think that the humectant in the product would really shine in drier conditions. Is that true?
So you mentioned humectant. So what we're talking about here is a product that is an enzyme type product, and there's several components to that. He mentioned one of them is humectant, which is basically a substance that attracts moisture from within the residue, the environment, and tries to hold it within that environment so that those enzymes can work. So that's one factor we've got in there. There's also some chelated nitrogen in there to help kind of as a food source for any of those enzymes and microbials that'll start working on this product. So there's two enzymes. There's endoglucanase and hemicellulase. The endoglucanase targets the cellulose in these stalks. So all this pith is some very tough, as everyone knows, long branch carbohydrates. So what that enzyme will do, that cellulase, will basically start to cleave that long chain carbohydrates and give you a lot of shorter pieces. What's the advantage of that? Well, now I've got shorter pieces. I've got multiple attack points now for both moisture, the enzymes, and microbials that are going to start breaking those down. It also gives more access points to the glucose. So eventually this turns into glucose too and releases some of those sugars. The hemicellulase will do the same thing. It'll take some of those polysaccharides and start to expose those to those live organisms. Those live organisms are the things that really end up doing the work on this, right? So we've got a lot of components to try to attack that. You talked about residue, right? It's trying to manage this. So you've talked about the nutrient cycling. You mentioned a little bit about stand establishment, and that's what we're trying to figure out too, right? So if we can get this broke down early on before the season, whether you've got an early strip till application or even at planting, that's what we would like to see with this product, which is a little different than the way we thought about residue decomposers in the past with some live organisms that we start in the fall and maybe go ahead and do the spring to try to get a long-term degradation. The advantage of these enzymes is that we can get them maybe that 2 to 3 week ahead of planting or at planting and hopefully get some advantages from a mechanical side also to help with that seed placement. So nutrient cycling and seed placement is kind of the targets we're trying to do with this product.
Absolutely. Now, for our trials this year, we're looking at spring applications, like you said, pre-plant and at-plant applications. I think we're really going to get a good idea of product efficacy because at V7 on corn and then at R1 on soybeans, we're going to go in and take tissue samples, soil samples, and residue samples and send those off to Midwest Labs. So I think that will paint a really good picture for us to figure out whether the product's actually cycling nutrients for us. And I think another interesting topic that we were talking about at lunch was trying this product in the fall. We're obviously doing spring this year, but you kind of brought that up in the fall. What would that look like?
Yeah, if we could get it, something done right after the combine, kind of to get that jump start and maybe get away from doing maybe a fall pass of some tillage or something like that. And, just kind of get ahead of the game because it's a game changer when we can get rid of that trash and get it into more of a manageable situation.
Yeah, and I think that's very viable, right? We're going to have some actual targeted testing for the fall to look at that. And I think your concept is right on. If we can, in the spring, we're targeting nutrient cycling early for that early plant and also trying to get that residue chopped up so that the machinery can do its job in the spring also. But then come fall, we're going to have more residue, right? So to your point, let's get a jump start on that. We're going to have that window in the fall where we still have some temperatures, we got some moisture, and we get an enzyme working on that residue. That's just a little bit farther ahead that we're going to be in the game come spring again.
Absolutely. Yeah, so I think it'll be a really key product, especially for high yielding growers like yourself. When you're getting over 300 bushel corn consistently, there's a lot of residue out there to manage. So I'm very excited about where things are going to head and look forward to the results.
Yeah, and I think the other thing to think about is we have, along with this product, is 2 products called BioPath and PowerCo, which are our live organisms. The advantage we're going to get early on, we hope, is once we prove the degradation of these products, we get those open cells, and what they're doing is they're opening themselves up to microorganisms. So a product like a BioPath or a PowerCo now has a better opportunity to cycle nutrients out of the residue along with the soil profile.
Yeah, I think that's a key point too. And I know you've hit on it before, but just to reiterate, the predictability from an enzyme-based product and also the fast activation, right? It takes biology to colonize that medium. That takes time, right?
Yeah. So you're kind of jump starting that process. You're going straight to the enzymes. In a microbial situation, those microbes are going to produce organic acids and enzymes. Here we're taking the enzymes right away. We're stomping them in there. That's going to narrow that window of activity because they do have a life cycle. But the hope is that within that 2 to 6 weeks, we get enough degradation to get that product concept delivered.